I'm happy to share with you this afternoon on the topic, the lethal yellowing disease of coconut in Ghana, the pathogen, hosts, and varietal trials. Well, firstly, the coconut is a very important cash crop along the coastal belt of Ghana. It is grown as a subsistence crop in the coastal area of Ghana. And in these areas, the soils are poor and most crops cannot do well, but the coconut thrives in this area. Coconut supports about 4.2% of the national population. About 4.2% are dependent on the coconut industry. Okay, and the products from coconuts are used by both rural and urban dwellers. So here we can see the husk being used as a source of fire in the rural areas, and the urban areas, a little bit more affluent people can afford high-end products such as the virgin coconut oil, which is becoming increasingly popular in the country because of its health benefits. The crop and the industry, however, is threatened by a lethal yellowing type disease. The disease was first observed in 1932, and so far it has destroyed about 20,000 hectares of palms in the country. Now, the disease symptoms are similar to what we have, the previous speakers have mentioned. It starts with the premature nut drop, and there is progressive yellowing of the palm fronds, starting from the lower and oldest leaf and progressing to the inner leaves. And at the terminal stages of the disease, there is what we call the characteristic telephone pole. All right. Now, the pathogens. The LY in Ghana is associated with group 22 ribosoma group, the subgroup B. In the group 22, there are two main subgroups, subgroup A and subgroup B. The subgroup A is referred to the Candidatus phytoplasma palmicola, and the B is the Candidatus phytoplasma palmicola related strain. Now, under the auspices of the Tropicsafe Project, we have sought to study the diversity of the phytoplasma that is present in Ghana. And to do this, we have undertaken surveys, extensive surveys, using accessible routes and footpaths to go around the whole country looking for disease symptoms. And we have also, in the process, sampled over 100 trunk borings using motorized drills to take phloem tissues in the form of the sawdust. And we have investigated the diversity of the phytoplasma using the Lucille tRNA synthetase gene and ribosoma gene-based PCR assays. For all positive tests, we have sequenced them. All right. Now, what we have realized from our results shows that for the 16SR sequences, for all the samples across the country, the sequences are the same. There's no diversity in the 16R sequences. But when we have used the Lucille tRNA synthetase gene, we have found two unique SNPs that differentiate the phytoplasma between the palms occurring in the Volta region of Ghana and those occurring in the western and central part of Ghana. So the strains that we have identified are geographically restricted. One just specific for the western and central parts of the country, and the other is restricted to the Volta region of the country. We have also sought to find out if there are secondary plant hosts that harbor the phytoplasma associated with the coconut disease. And to do this, in the Tropicsafe Project, we have sampled about 24 plant species belonging to 18 plant families from within and around affected coconut plantations. We have done the survey in the three main coconut growing regions of Ghana, that's the Western, Central, and the Volta region. And we have also done the surveys and the sampling, both in the rainy season and in the dry season, to take into account of the effect of the climate on the different crops that occur in both seasons. We have done the detection and identification of the pathogen using PCR and sequencing. And this is a map of the area where we have sampled, that is from the Western up to the Volta region of Ghana. we have identified from our survey and analysis 3 plant species that are known to harbor the phytoplasma associated with the coconut disease. The first is Stachyphyta indica. Then another plant we have identified is a Pathogen, Petandrus, and then Lapotia esuans. And this is the first time we have identified secondary hosts harboring the phytoplasma associated with the coconut disease. On insects, the vector of LYD in Ghana is unknown. So under the auspices, again, in the Tropicsafe project, we have carried extensive sampling of insects, suspected possible vectors in devastated areas, in areas where the disease has already passed, in diseased areas, and in LY-free areas. And we have also carried out the sampling in the Western and Central regions. And we have done it both in the dry season and the rainy season. We have collected over 2,000 samples and analyzed them using nested PCR and sequencing. And this shows the areas from where we have collected the samples. We identified 2 species of Patara, that is the Patara amata, and 2 other species of Distrombus, which are known, which we identify them to harbor the phytoplasma. As a result of the exigencies of the project, we were unable to do the transmission trial, but hopefully we should do the transmission trials in future. Another important leg, as we have learned from the previous speaker, in managing the later yolling disease is to grow tolerant or resistant coconut varieties. And to this end, under the Tropicsafe project, we established a varietal resistance and agronomic trial using 5 promising dwarf coconut varieties that have been tolerant to the disease in previous trials, in a trial that started in 2007. In Ghana, the most acceptable the variety to the later yolling disease is called the West Africa tall ecotype. And we have included the West Africa tall ecotype, and the current hybrid that is given to farmers, we call it the SGD by VTT. We have included them in the trial. And the trial has been established in a diseased area in the western region of Ghana. We started in 2017. So the ongoing activities, we are undertaking data collection, looking at the vegetative parameters, various vegetative parameters. And then we also started the palms are started flowering and bearing. So we are collecting floral data and floral emergence and floral data. All right, preliminary results. So far, we have realized that from the analysis, there are 4 varieties that have shown significant superior growth compared to the hybrid that is currently being distributed to farmers. And this includes varieties such as the NLD, IBD, NGBD, and the West Africa tall, which is a susceptible variety. So we are not too much interested in that. On our expectation, the expectation is that through the Tropicsafe project, we'll be able to recommend new varieties for planting, for replanting the coconut industry, coconut farms in Ghana. And we also want to diversify the planting material base for the country, so that, as the previous speaker mentioned, we need to have a number of varieties on a farm, and that helps to defeat the disease. Post the project, we aim to do fruit component analysis, proximate analysis, and also look at the Brix level of the various varieties that we are trying. So in summary, through another Tropicsafe project, we have identified 2 strains of the group 22 phytoplasma, and this will influence our future resistant trials and how we manage the disease in the country. We also have identified plant hosts, now 3 species and 3 insect species. We will incorporate the alternative hosts we have identified in the management of the disease. We'll remove them so that it slows down the spread of the disease from farm to farm. And the insects will be included in future transmission trial. We are also hopeful that at the end of the project, we'll be able to make a recommendation on varieties that will be included in the breeding program in Ghana. Thank you very much, and I look forward to answering any queries that you may have. Thank you.